On to part 3 of chapter 15. So in 15:4, there's a discussion of the alterations of the number of chromosomes and the structure of those chromosomes causes genetic disorders. So we're going to take some time to look at some of the genetic disorders. So when we have large-scale chromosome alterations in a human and other mammals, often that's going to lead to a spontaneous abortion or miscarriage before an embryo or a child is actually born. If the child is born, it will cause a variety of developmental disorders. Now, plants are going to tolerate genetic changes much better than animals do, but some possible errors that can occur in both plants and animals include an incorrect number of chromosomes. And this results from a couple of different things. One of them is non-disjunction, which means the chromosomes don't separate properly during meiosis. But we can also have the breakage of a chromosome. And we can have what we call different types of mutations that are either deletion, duplication, inversion, or translocation. So here are some more specific examples. In non-disjunction, we have pairs of homologous chromosomes who do not separate normally during meiosis 1. The result is that one gamete is going to get 2 of the same type of chromosome, and the other gamete is going to receive 0 copies of, so that one of the homologous pairs will go in with the other, and we end up with another gamete that's totally missing that chromosome. And this is usually the result of a problem with the meiotic spindle in terms of how this error occurs in the daughter cells. So you can see here the results, a very simple drawing. This is another more detailed drawing of non-disjunction. So typically, we have the 2 homologous chromosomes here separating, but with non-disjunction, here's one that should have been going this way, but it's not. It's going to go together with its homologous pair, its homolog. And the result then is here is the separation in meiosis 2 of this one set of sister chromatids. And here we have, where we're supposed to have just 2, we have actually 3 sets of sister chromatids separating. But even in meiosis 2, during anaphase, we can have another non-disjunction occur. We can actually have the sister chromatids not separate. So all of these non-disjunctions, whether it's meiosis 1 or meiosis 2, result in a chromosome with the wrong number of chromosomes. A cell with the wrong number of chromosomes in it. Here we have this one where we have 3 instead of the 2 we should have had. And in this one we should have had 2, but now we only have 1. And here we end up with some normal because this disjunction occurred in meiosis 2. So it's only occurring right here where we should have had the separation of these chromatids. Instead, we end up with just that one lonely one there, and then 3 over here. This is a video showing non-disjunction in mitosis. So hold on a second and we'll show that. So you can see here the different phases of meiosis occurring, and see if you can identify the different phases. That obviously was anaphase and telophase and cytokinesis. Okay, so non-disjunction results in what we call aneuploidy. And so when a baby, for example, gets the wrong number of chromosomes, that's a form of aneuploidy. And so trisomy is when, for example, a cell can have 3 copies of a chromosome, or monosomy when cells only get 1 copy of a chromosome. So you can actually look at this and follow through, and pause it if you need to think through this and examine the difference between the two. But this is in contrast to what we call polyploidy. polyploidy is a different situation because it's a condition in which an organism has two complete sets of chromosomes, so more than two of the complete sets. So, for example, we can have triploidy, which is 3 sets of chromosomes, or tetraploidy, which is 4 sets of chromosomes. So polyploidy is actually very common in plants, but it is not common in animals. And polyploids are much more normal in appearance than an aneuploid. So an aneuploid is when, again, we have the wrong number of just a single chromosome. But a polyploid is when we have a complete extra set. So looking at some examples of these situations. In the Turner syndrome is a type of genetic disorder. It's a monosomy disorder, and it's called monosomy X. And it occurs 1 in every 5,000 births. And there are different degrees of effect, from a webbed neck to short stature, and sterility is actually also part of this. So in this case, you either get a monosomy X or an XO. So what does that mean? Hopefully that is something that you can figure. It's that you only end up with 1 of the X chromosomes instead of 2, and you're a female. Down syndrome is an aneuploid condition because one of the sets of chromosomes, chromosome 21, ends up with an extra chromosome. So it's called trisomy 21 because as you look at this karyotype right here, chromosome 21 is the last of the chromosomes right here, one of the last. And so it's a very small chromosome. But you can see here, instead of having 2 homologous pairs, there's actually an extra one there. So that makes it a trisomy. So there are 3 copies of this chromosome 21. And this occurs in 1 in 700 children born in the U.S. And it has very severe effects. In this syndrome, some of the defects, actually many of children, more than 80% of those with this condition die within the first month of birth. But those who don't actually have to deal with some pretty severe problems like seizures or sleep apnea, deafness, eye abnormalities. It's amazing how many things get tied into having this extra chromosome. Most infants with Down syndrome have a cleft lip, a cleft palate. They have congenital heart disease. They get hernias. They have genital abnormalities. So even though this is a pretty common syndrome, it is a very significantly damaging syndrome. There is a correlation between Down syndrome and the age of the mother. So this chart actually shows you that as a woman gets older, the chances significantly go up, from 1 in 1,000 if you're under 30 to 1 in 12 if you're over 49. So this makes a lot of sense because as the age of the mother increases, that means her eggs that went through meiosis 1 before she was born are getting older. And so the chances of them going through meiosis correctly decreases with time. So human chromosome disorders are pretty common in humans, but we don't see them because most of the embryos with them are spontaneously miscarried or aborted. Because the alterations that occur are too disastrous to a healthy life. And I'm actually going to come back to that. So there are some things that are abnormalities that occur just on the sex chromosomes. human development is actually more tolerant of the wrong number of sex chromosomes, which is kind of interesting. But there are a variety of distinct syndromes in humans, and so these are 4 of them. So we already talked about the Turner syndrome, where the female is missing one of the X's, but there's also what we call Klinefelter's syndrome in males and Jacob's syndrome in males. And you can see what happens here. In Klinefelter's, the male gets an extra X. In Jacob's, it gets an extra Y. And in trisomy X, the female gets an extra X. So Klinefelter syndrome, 1 in every 2,000 live births have this. They have male sex organs, but they are sterile. And there can be feminine characteristics, like breast development or lack of facial hair. They tend to be tall, and they tend to have normal intelligence. So that's when males get an extra X. And you can see again, down here, you can see the karyotype showing that extra X chromosome. And here's another close-up of that extra. This one actually, this male here has 4 X chromosomes. On the contrast with Jacob's syndrome, the male ended up with 2 Y chromosomes. You can see that right here. That extra Y chromosome results in some symptoms like a slightly taller person, more activity in that person, maybe some slight learning disabilities, or a delayed emotional maturity, but things like intelligence and sexual development are normal with Jacob's syndrome. And then with a woman with an extra X, there actually is no outward symbol or symptom that this has occurred. And the reason seems to be because of the formation of Barr bodies, which again is that X inactivation that occurs. So instead of inactivating just 1 X chromosome, the body can actually inactivate 2 of the X chromosomes. So it seems to not be a problem at all, trisomy X. So ways to figure out whether someone is going to have any sort of genetic disorder is through prenatal genetic testing, prenatal being before birth. And it's done through amniocentesis in the 2nd trimester. And they can take a sample of the embryo cells, and then they can come up with a karyotype in that way. And through that karyotype, you can certainly tell if you have an extra chromosome in here. You can see some of the missing chromosomes or non-disjunction that's occurred. There are also structurally altered chromosomes. And so these occur from different types of mutations. For example, there's a syndrome called cri du chat, which is a cry of the cat. And that comes from a deletion in chromosome 5. That means part of chromosome 5 has been removed. And a child born with this syndrome is severely intellectually disabled and has a cat-like cry, and they usually have a very short lifespan. There are also cancers like chronic myelogenous leukemia, or CML, that are caused by translocations of chromosomes. And a translocation is when one part of a chromosome trades place with another part. So this is an example of what that would look like. So you see here normal chromosome 9 and normal chromosome 22, and they exchange the wrong, they're not homologous, so this is not crossing over. They actually exchange portions as if they were crossing over, but they're the wrong genes going to the wrong chromosome, and they result in a form of leukemia. So these are 4 of the types of chromosome alterations. There's deletion when you remove a chromosome segment. There's duplication when you repeat a segment. There's inversion when you reverse the orientation of a segment, or translocation when you move one segment from one chromosome to another. These are pictures showing that. You can pause and take a look at this. And finally, you can see here another picture showing the different types of chromosome changes that result in chromosome abnormalities or genetic abnormalities.